Hello everyone, welcome to the Science Stuff. Today we are going to be learning about the basics of nucleic acids. So what are nucleic acids? Well, let's find out. Now, nucleic acids are one of the four major macromolecules essential for our bodies. They are long-chain polymeric molecules consisting of single units or monomers called as nucleotides. They are organic molecules, and because of this they are obviously made up of carbon, hydrogen and oxygen. And along with this they are also made up of nitrogen and phosphorus. For high school, the two main nucleic acids that you need to know about are DNA and RNA. Now let's look at the basics of DNA. DNA stands for deoxyribonucleic acid. DNA is composed of a pentose sugar, a phosphate, and some cyclic bases containing nitrogen known as nitrogenous bases. Now the phosphate, the sugar, and the nitrogenous base combine together to form one nucleotide, which is the monomer that forms the DNA. Now these nucleotides continuously repeat to form this beautiful structure. Now let's take a closer look at one nucleotide. Here we can see that the sugar contains a 5-carbon chain. Now where is the nucleotide found, or where is the DNA found? DNA is found in the nucleus of eukaryotes and in some organelles like mitochondria and chloroplast. Now what about prokaryotes? Where is the DNA in prokaryotes? Well, in prokaryotes, the DNA is contained in a central area of the cell called the nucleus, which is not surrounded by a nuclear membrane. Now let's take a look at nitrogenous bases. There are 4 nitrogenous bases in DNA: adenine, guanine, cytosine and thymine. Adenine and guanine are purines, while cytosine and thymine are pyrimidines. Adenine combines with thymine with 2 hydrogen bonds to form the rung-like structures in DNA, and guanine and cytosine do the same thing but with 3 hydrogen bonds. DNA is important because it contains the hereditary blueprints of all life. An important factor of this is nitrogenous bases. They function as the fundamental units of genetic code. Every single person is different because the sequence of nitrogenous bases is different in every person. So say if we take the same chromosome, say in humans chromosome number 21. Now this is just hypothetical, but in that chromosome and in that specific DNA, if the nitrogenous bases were adenine, thymine, guanine, cytosine, adenine, thymine, thymine, adenine, guanine, cytosine, and in another person's DNA they were thymine, adenine, thymine, adenine, guanine, cytosine, cytosine, guanine, basically it's different. So the order of the nitrogenous bases is different for every single person, making us fundamentally unique. Now let's look at the anti-parallel nature of DNA strands. The anti-parallel nature of DNA strands is needed to form the hydrogen bonds between the nitrogenous bases. The nitrogenous base connected to the 3' carbon connects to another nitrogenous base connected to a 5' carbon in the other ladder rung. The anti-parallel nature makes the DNA stable by facilitating the formation of more hydrogen bonds in the nitrogenous bases. Now let's take a look at RNA, the other nucleic acid. RNA stands for ribonucleic acid. Ribonucleic acid is single-stranded, as you can see right over here. RNA is important for protein synthesis, and it has uracil instead of thymine as its nitrogenous basis. The other three are the same: guanine, adenine, cytosine, and instead of thymine, it has uracil. Now, RNA also serves as the primary genetic material for viruses. Now, as you can see here, these green color things in this virus are RNA strands. Now, RNA strands and DNA strands are very similar and have very minute differences. The pentose sugar is different. In RNA, it's ribonucleic acid, so ribose, and in DNA, the sugar is deoxyribose. Here you can see the main difference between ribose and deoxyribose. The deoxy refers to the lack of an oxygen in the 2' carbon atom. So if this is 1', this is 2'. So while ribose has a hydroxyl bonded, deoxyribose only has a hydrogen bond. And this is the difference between their pentose sugars. So what are the types of RNA? There are three main types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Previously, we established that RNA is important for protein synthesis. These types of RNA work together to synthesize proteins. Now you'll learn more about this in the chapter on gene expression and regulation. Thank you for watching this video. I hope you now have a clearer understanding on nucleic acids. For more educational videos, please like, share and subscribe, and comment down below any questions or suggestions you might have. And remember, you're watching The Science Stuff.